Hey guys, so welcome to chapter 3.1 of MCAT biology, and today we're going to be going over prokaryotic cells. So last video we went over eukaryotic cells, and now we're going to go over prokaryotic cells. So a few things. Prokaryotes are the simplest of all organisms. They simply only have cellular DNA in a nucleotide region. So you can find the DNA in that region. So even though that they're very simple, there's a lot of different types of prokaryotes. So let's get into it. So there's really lots of different types. There's two different types, but if you get really specific, right? So archaea is our first one. So what you really need to know about archaea is the most important thing is they really know how to live in an extreme environment. Hint, hint. I'm not saying anything about a test or whatever, but it's not like I've seen that before. But definitely know that these archaea live in bad environments, right? So they can survive high temperature, high salt, high chemical. That's pretty much the most important part. They are very similar to bacteria, but they are a lot closer to eukaryotic cells than they are prokaryotic bacteria. So next, bacteria. So bacteria are prokaryotes. As we know, they're single-celled organisms. They have no membrane-bound organelles, and they make up a large proportion of the amount of living organisms on the planet. A few things that you need to know bacteria is, first off, their shape. So you can have either a rod shape, a circular shape, or a spiral shape. Your bacteria could show up. If you take microbiome, you will see this pretty cool. But yeah, these are the basic bacterial shapes. Nothing much here. This is just more for you guys to understand how these things look. So cell wall, bacteria, we're going to go into more detail down here, but bacteria have cell wall, which in most cases are they're a target for antibiotics. So flagella, this is also pretty important about bacteria. So you can look at flagella as if it's like a motor of a car. So the bacteria uses flagella to really propel it. Okay, like it really moves it. So yeah, it really, like, if let's say that I can ask you a question about a bacteria who had a mutation of a flagella, you should know, like, okay, the way you think through it is, okay, wait, flagellas help with motion, so I'm going to assume that this bacteria can no longer move. So this bacteria is going to be non-mobile. And then pili, again, they're just hair-like structures, and this is just a way to form genetic transfer. So by conjugation, think of it, now don't draw this in your notes, but it would look something like this, right? And then you'll be crossed through the conjugation bridge. So again, this is how you transfer genes. So yeah, so back to here. So again, definitely just know these three shapes. I haven't got any specific questions about what is this, what is that, but this is, in my opinion, very basic and something that you should know to better understand other stuff. So we need to go through the difference. This is the last big difference about bacteria and eukaryotes. So there's two different types of bacteria cells. So you have gram-negative and gram-positive. So gram-positive has a very thick peptoglycan layer. Now, peptoglycan sounds like a big word, but just think of like a very thick layer of protection or something, right? Right. So peptoglycan, you could break that down. It's like peptide, but definitely know the difference between the two. So gram-positive is thick, but gram-negative have a thin peptoglycan wall, and it stains pink. So you need to know that gram-positives, they stain purple. Gram-negatives, they stay pink. Gram-positive has a thick layer, while gram-negative has a thin layer, right? And the outer membrane of a gram-negative contains what we call a lipopolysaccharide. Okay, so that's the main difference, is gram-positive, thick peptoglycan wall, gram-negative, lipopolysaccharides. One more thing about gram staining. Again, we already went over this a little bit, but gram-positive retain the crystal violet stain, and that's due to the thick peptoglycan wall. So you do this a lot in microbiology lab, but that wall pretty much absorbs the stain. Versus gram-negatives, they lose that violet stain. And then what you would do is you would then use like a pink color as a counter stain. So I see you guys in the next one. That's the end of this video. Hopefully that was a little helpful.